Hey everybody, it's Mike Jones again. This is Biology 121, Anatomy and Physiology, first semester number 1 at Grand Rapids Community College. Today we're going to get into bioenergetics. Bioenergetics is a long word for the production of energy in cells. Cells require energy to do their work, like everything in life. Right? Your car needs energy. It gets it through gas. You need energy. You get it through food. We're going to talk about that specifically, right? Your phone, your house lights, everything, computer, it all needs energy. It gets energy in its various forms. Cells need energy to do the work that cells need to do. This is how it does. This is how it's done. The thing that you're going to see over and over and over and over again is ATP, this word, adenosine triphosphate. We're going to talk about its specific structure and why it's so important. It is cellular money. Now, I've got to get my pointer. There's my pointer, and we've got to get on to the next slide. So you have the slideshow available to you. It's in the Unit 2. Trying to think of what folder it's under. Resources, I think. Resources? Yeah. It's where you should have gotten a copy of this video as well, which will be on YouTube, of course, for you. So, we got to talk about some terms, of course. Everything in science has terms, has a definition to it. In this case, we're talking about energy. What is energy? Energy is the capacity to change something, the ability to do work. Energy is necessary to do work. So we have, of course, common forms of energy that you're quite familiar with. Heat, light, sound, electrical energy, mechanical energy, chemical energy. The body works mostly with chemical energy, but, of course, light is in the eyes and sound is in the ears. And these different forms of energy have to be changed into an understandable format. And that's allowed. Physics allows that to happen. And so the physics of the situation is that energy can't be created or destroyed. You can't just like poof, create energy. You have to change its form in order for it to be useful to you or to a power plant or to a rocket or whatever. In a cell's case, what a cell does is it produces energy through what's called cellular respiration. And there are a couple of different parts to cellular respiration, but it's called respiration. Yeah, you're going to think of respiration as breathing, and that's kind of it. Part of what you think in the vernacular, what you think of respiration is partly true, but respiration is bigger than just simply that, taking a breath in and breath out. There's more to respiration. So cellular respiration is taking resources from the environment and converting it into a form that the cell can use. Right? And so most metabolic reactions in the body use chemical energy, which is understandable. All right, so I told you ATP. ATP is cell money, adenosine triphosphate. There is something particular. I can't tell you exactly why it is that ATP, why the universe or why Earth biology chose ATP, but it did. And so ATP has that special place where it is cell money. And when I say it's cellular money, what I mean is it is the currency with which a cell can use in exchange for doing things, actions, activities that the cell requires to be done. Now, when I say cell money, I mean if you're in the United States, which most or all of you are, if you live here, the money that we buy things, if you go to the store, if you go online, right, you need US dollars. Now, you may come from Japan and you have yen, you have Japanese yen. Or you come from Russia and you have rubles, and you bring that money, that's money there. You bring that money here, and if you go to a retailer and you say, hey, here's my Japanese yen, they're going to say, that's no good here. Of course, it's money. You can exchange that money at a bank or some financial institution. You can exchange it for US dollars, and then you can buy the things that you want to buy. Here, that's adenosine triphosphate. Adenosine triphosphate is the money, is the energy source of choice by the cell. Now, what does that mean? Well, we have to convert anything that we take into a cell has to be converted into ATP. So that means, and I mean literally, anything that you eat, if you eat a bagel, if you eat a steak, if you eat vegan, I don't care what goes in your mouth, it has to be converted on the cellular level to ATP in order to get any work done. Otherwise, it's a waste of time. Literally. So adenosine triphosphate is where it's at. Adenosine triphosphate is composed of these three parts. Now you're going to see that this, the adenine group here, you already went through DNA, and you know DNA and RNA have adenine as a base. Well, here it's put to different use. Right now you have a ribose sugar. Well, that's like RNA. Yep, it is like very much like RNA. However, the big difference here are these things out here, these phosphate groups. The phosphate groups are referred to as high energy phosphate groups. And in particular, the bond right here between that second phosphate and the third phosphate, this is considered a very high energy bond. And that energy that is in that bond is the energy that is used, converted from this chemical energy into actual work energy to do other things that the cell needs to have done. So we refer to this as a high energy bond. I like to think of that, the second and third phosphate, as elementary kids or middle school kids on a bus. It's a crowded bus. There's two friends that are sitting in a seat, but three kids could fit on that seat. Third kid comes in. There's no other seats available. He's got to sit on that seat with the two other kids. Right? The two other kids are these first two phosphates. They're friends. They're happy. They like each other. They want to sit next to each other. This third kid comes on. They don't like him. He's a jerk. They just don't like this kid. And so that kid has to sit on that seat, and there is tension there immediately. Now he's got to sit there, and they have to let him sit there. But nobody's having any fun here. Well, that's the second and third phosphate here. There's a lot of jostling. There's a lot of energy there. And it needs to be released, but it needs to be released into usable, into a usable form and in discrete quantities so it can be used efficiently. Right now, the body constantly, and I mean constantly, creates and uses ATP molecules. And what happens when ATP is used? Here's ATP, so we're going to pretend we've already formed ATP, we've already made ATP. We've got these high energy bonds, or the high energy bond out there between the second and third phosphate. Great. Right? Now we take that phosphate off, but in doing so, that releases that energy that's held in that bond. That's the energy that can be used for other metabolic reactions. So if you're going to break this down, that energy is going to be used to build something else up. Right? So you're going to use it in anabolic reactions. Right? You break this down, which is a catabolic reaction, and you harness the energy and put it into anabolic reactions. But now you have a molecule that is now called adenosine diphosphate. Only has two phosphates out here. Well, you got to shove another phosphate. You got to put this kid, a third kid's got to go on that bus seat. So you shove that phosphate on there. Well, yes, that takes energy to push that phosphate on there. So you're constantly cycling back and forth between ATP, ADP, taking the energy and using it in other forms and other reactions, but you got to recreate the energy. So this cycling goes on and on and on. Right now, when you release chemical energy, The interesting thing about ATP is you would think, okay, well, we're going to take that energy and we're going to put it into some other reaction. We're going to use that energy. It's like a power plant, right? The power plant makes energy and it releases that energy out to you as electricity, and you charge your phone and you work your computer with it. But not all of that energy that they produce comes to you. Some of it gets wasted along the way. Now, in their case, some of it gets wasted on the electrical power lines where the electricity is not 100% effective or 100% efficient in coming from the power plant to you. Some of it is lost just simply into the wire itself. Well, in your case, in the case of cells, in the case of ATP, it turns out that when you release that terminal phosphate, that third phosphate, when that comes off and you get the energy, 40% of that energy is released as chemical energy, and that's what you can use to power other reactions in the cell. But the other 60% of that energy is released as heat. And you say, my gosh, what a waste of energy. Not at all. That heat is what makes you a homeotherm. Homeotherms means that you create your own heat. Right? You're creating an internal furnace. So does your dog, so does your cat. All mammals do that. And that's what distinguishes mammals, is that we create our own heat source. That's why we have a temperature, right? Our normal homeostatic temperature as human beings is 37 degrees C or 98.6, or, you know, it's a range like all homeostatic mechanisms. Dogs are a little warmer than we are. Turtles are a little colder than we are. You know, it's but the release of the chemical energy from ATP, 40% is used in reactions, 60% forms your internal furnace, which is absolutely essential as a human being to have that, or as a mammal. So that heat energy comes from ATP. Now, what does that mean? That means on a physical level, if you are cold, get up, move around. If you start walking, if you start exercising, of course you get warmer. Why do you get warmer? Because you're using ATP. 40% goes into keeping you moving around. The other 60% keeps you warm. That's how it works. All right. I told you it's called cellular respiration. Well, the process of cellular respiration, the process of taking food, whatever you eat, it's digested down to its monomers, right? Amino acids in the case of proteins, monosaccharides in the case of sugars. What am I forgetting? Fats don't have a particular monomer, but you do still have to break them down. But these sources of energy to you are broken down in a specific process. And what we're going to get into is the preferred source of energy for all of your cells is glucose. Glucose is a monomer, is a monosaccharide, 6-carbon sugar. It turns out that this is the easiest sugar for your cells to turn into ATP. Now, there are ways that your body has to turn like amino acids into glucose. It can turn lipids into glucose. It can turn nucleic acids into glucose. It can convert all of these things. There are ways to do it. There are clever ways. If you take a more advanced biochemistry course, you're going to learn all about those ways. For our purposes, we're not interested in all that stuff. All you have to do is to think, I'm going to start with glucose. If the cell starts with glucose, which is the preferred energy source, now, how do I get to ATP? That's what this process is all about. 3 steps to it. 3 huge, big steps. The first one is called glycolysis. The second one is called the citric acid cycle. The third step is called the electron transport chain, and that is connected to another way of making ATP called oxidative phosphorylation. Now, I want to point out something to you. You see, glycolysis says anaerobic after it. This is a term that means without oxygen. It does not. This is a process that does not require oxygen, doesn't need oxygen. It will continue without oxygen around. Anaerobic bacteria live by glycolysis alone. They're perfectly happy with that. If you were a single-cell organism, you could do the same thing. Unfortunately, you have trillions of cells, and they're all demanding energy. So you need the next steps, right? You need the aerobic steps. So the citric acid cycle is aerobic. Electron transport chain oxidative phosphorylation is also aerobic. You must have oxygen for these things to happen. No oxygen is required here, but you've got to have oxygen for the next two steps. What's the difference here? The difference is glycolysis, and you're going to see this in numbers. Glycolysis is a fairly inefficient way of producing ATP. You'll get some ATP, and as I say, bacteria get enough. They're fine with the amount of ATP that they get out of glycolysis alone. You're not. You must have the aerobic parts. That's why you have to continuously breathe. That's why you have a circulatory system, to get oxygen to your cells so that they can produce their own source of energy. All of these things. Now, why do we bother? Why do cells bother going through this process? They bother because you can release energy in two big dramatic ways. You can release energy all at once. Now, you've already seen a video about this where I showed you if you have solid sodium and you throw it into water, it wants to lose that outer shell electron very, very rapidly. And it does that. And when it does that, boom, you get an explosion. So that is one way to release energy or to harness energy very, very rapidly as a bomb. You release it all at once. That doesn't work for biology. So what biology does, what cells do, is they have these other two ways, the citric acid cycle and electron transport chain. These are ways to take bits of energy out in smaller doses and then convert it to ATP in smaller doses. And that's really what they're all about. And so you don't blow up. That would be bad for you. Like I need to tell you that. These other ways of extracting energy, extract them in smaller, tinier amounts. And so that's why you have all of these steps. Now you're going to see there's a whole bunch of steps to this process, and it's going to blow your mind, and you're going to go, why do cells do this? They do it to frustrate you, of course, in your learning process. No, they don't do that. They do it to extract energy in small amounts, smaller amounts, and then convert it to ATP, which is the usable source. So that's what these are for. All right, now cellular respiration. You go through glycolysis. I'm gonna show you the important parts, the parts that I want you to know about. So glycolysis starts with a molecule of glucose. Tattoo that on your arm, put it on your forehead, I don't care, sleep with it under your pillow, whatever. Glucose. You're starting with the molecule glucose, a monosaccharide. Now, if you have table sugar at home, you have glucose. But what you have is you have glucose and fructose hooked together. That's what table sugar is. Table sugar is actually called sucrose. And sucrose is a disaccharide. In order for it to be useful to you, you have to chop those two monosaccharides apart from each other. Then you get glucose and you get fructose. And fructose can be converted into glucose and then used in these reactions. All right. So you go through this first part of glycolysis. No oxygen needed. We're going to talk about the specifics of that in a moment. But the next step is this process that is referred to as the citric acid cycle. Now, we'll get to the specifics of the citric acid cycle in a moment. But what you have to look at here or realize is it's a cycle. What's a cycle? Well, that means that something goes in, some substrate goes into this cycle. And the cycle is already going. Don't worry about how it got started. It's already going, right? It's cranking away. Round you go. And a cycle simply means you put something into the cycle, and then it turns into a substrate. And then something happens to that substrate, and it turns into another substrate. Now, actually, it turns into a product, but the product is the substrate for the next reaction. And that product is the substrate for the next reaction and the next. And around and around and around and around and around and around you go. And, you know, I know it says you get a product, but you don't really get a product per se. What you're getting is this cyclical arrangement of molecules and conversions. And the whole point to a cycle is to extract energy in small doses that you can convert to ATP someplace else. That's the point. So, an example of this, of course, is the citric acid cycle. And we're going to see, and that requires oxygen in order for it to continue through the cycle. Now, I do want to show you something. Let me pause this for a moment. All right, I'm back. You don't realize that I left, but I was in the slideshow, and I thought this is a picture that I put in the Module 2 folder, or the Unit 2 folder, to describe all of the reactions, so the sort of the general overview of glycolysis, citric acid cycle, and the electron transport chain, oxidative phosphorylation. This is the big overview picture. And you have this available to you. What this picture is telling you or showing you is you start with glucose. All right, so we've already said that. You start with glucose. Now these couple of arrows up here, glucose is not always immediately available from your intestines, right? You don't always eat glucose and digest glucose and put it into your bloodstream. Where you could get glucose, if you don't have any in your intestines to digest, you could get it from glycogen. Glycogen is a storage form. Now, glycogen is a polymeric form, a polysaccharide of glucose. So it's glucose molecules that are shoved together into long chains, and these long chains are stored primarily by the liver, but they're also stored in other places. Muscles also have a supply of glycogen. Liver is the primary place. So you can store glucose in a long chain format for use whenever you need it. But ultimately it's converted to glucose. Okay, so now glucose goes through this process. We're gonna talk more specifically about this. But the general overview here is that glucose is going to go through a process where it's converted into this product called pyruvic acid. The importance here is glucose has 6 carbons in a row. It's a 6-carbon sugar. Pyruvic acid only has 3 carbons. It turns out what happens is that you convert glucose into 2 molecules of pyruvic acid. So chemistry requires that you balance out all of the molecules. All the molecules that go into a reaction have to be accounted for at the end of the reaction. In the process of changing glucose to pyruvic acid, which is glycolysis, as it says right there, that is glycolysis. That is an anaerobic process, does not require oxygen, does not use oxygen. You get ATP. Some ATP. We're going to do some math a little bit later. I know, don't run away. Don't freak out yet. We're going to do some math. But you get ATP out of that. All right, so great. You get some energy. Well, as I said, if you're a bacteria, that's fine for you. That's all you need. You get a little ATP, that's all you need. You just crank through some glucose to pyruvic acid, off you go. Everything's fine. You're not a bacteria. You're a human. You need more energy. How are you going to get it? Now you go in the aerobic pathway. That's what this is right here. Aerobic pathway. When you go down the aerobic pathway, let's also define too glucose to pyruvic acid, which is called glycolysis. Glycolysis happens in the cytoplasm of the cell, or what we call the cytosol, the fluid portion of the cytoplasm that is outside the nucleus and outside the mitochondrion. I say mitochondrion on purpose because now we're going to get to the aerobic pathway. If we go to the aerobic pathway, we have to change pyruvic acid. Pyruvic acid can no longer be pyruvic acid. It has to be changed. And it's changed into another molecule called acetyl-CoA. That stands for acetyl coenzyme A. That's just the name of it. I tell classes every year, I don't name this stuff. All I simply do is I tell you what's going on. But acetyl-CoA now is inside the mitochondrion. In fact, it's inside the inner aspect, what's called the matrix of the mitochondrion. It's inside the matrix of the mitochondrion. This is the stuff that can now get into the citric acid cycle. And I told you, we went through kind of the example of what a cycle is. This stuff can enter into that citric acid cycle, and round and round and round and round and round it goes. And I told you the reason to do that is to either get ATP or acquire something, make something that can be converted into ATP. And so both of those things happen during the citric acid cycle. Something else that you'll notice during these aerobic pathways is you also generate carbon dioxide. So you're taking oxygen. Now, it's not the oxygen that's converted to carbon dioxide, but bear with me for a moment. You're breathing in oxygen and you're breathing out carbon dioxide. And this is where the carbon dioxide is coming from, the aerobic energy generating pathways. So you generate some carbon dioxide, making acetyl-CoA. You generate some carbon dioxide from the citric acid cycle. That's where carbon dioxide comes from. Then you send the products of the citric acid cycle, and we're going to talk about what those are. They go into this other process that's called the oxidative phosphorylation system. And that's where the electron transport chain is. And I'm going to tell you just briefly or basically, what do we mean when we talk about electron transport system or cycle? We're going that direction. But this is the big overview of how do we get energy from glucose. Now, there's another process that's illustrated here, and this will be a test question. I mean, I just pretty much guarantee you, if you don't have enough oxygen available, if you do not have enough oxygen, what happens to pyruvic acid? You didn't need oxygen to produce pyruvic acid, but you do need oxygen to continue through these other cycles. Well, what if you don't have enough oxygen? If you don't, you continue on an anaerobic pathway without oxygen. And you convert pyruvic acid to lactic acid. Now, you've heard about lactic acid. I hope you've heard about it. If you've ever exercised, if you've ever been late for an airplane flight or a bus departure, or you exercise, or you run up and down stairs real fast, whatever the situation might be, you're late for class, you start breathing hard. Why are you breathing hard? Because you ran out of oxygen. If you didn't have enough oxygen, now you get this lactic acid. Lactic acid is another way to produce a little bit more energy from pyruvic acid. You don't get a lot of ATP out of that, but you do get a little bit. But lactic acid creates this kind of muscle burn sensation. And it is not, you cannot continue to produce lactic acid and continue to live. At some point, the lactic acid will inhibit other enzymatic reactions, will slow energy production down, and you can't continue along those lines as a mammal. Again, as a bacterium, you can do it. As yeast can do this. Yeast have another way of producing a little bit more energy. They actually produce ethanol as a byproduct of their production of ATP. Well, that's good for us, right? We like ethanol. So we like the yeast to produce ethanol, but that's no good for the trillions of cells in your mammalian body. So this is the overview. Now we're going to go back to the other slideshow with this whole idea in mind, and how we go pyruvic acid to acetyl-CoA into the citric acid cycle. We generate some stuff that's useful for the electron transport chain and oxidative phosphorylation. Why? This stuff, ATP. We want to make ATP. That's our goal. All right. I'm going to stop this again. Back to the other slideshow. All right, so you've now seen the overview. Here's cellular respiration. Here's the example of citric acid cycle. Let's get to the specifics now. All right? Okay. Cellular respiration, we've talked about it. Here it is in total. Here is the big overview inside a cell. Now, you have a cell membrane on the outside, of course. Now, in this particular cell has a ginormous mitochondrion. Yeah, of course, cells have mitochondria are much smaller than this, and cells have lots of them, lots of smaller ones. Well, this is for demonstration purposes. So we start with glucose, 6 carbons. As I said, chemistry requires you to pay attention and to account for all of these molecules. Glucose goes through the glycolysis, and it produces 2 pyruvic acids. Pyruvic acid is 1, 2, 3 carbons. You start out with 6. You got 2 of these now. Great. The other thing that you got out of glycolysis was a net gain of 2 ATP. When I say net gain, I mean that's how many you produced overall. It turns out that you actually have to put in 2 ATP to get glycolysis to get going. So it's like priming the pump. You got to prime the pump. 2 ATP go in, and it turns out that in the end, you get 4 ATP out. So if you put 2 in and you got 4 out, 4 minus 2 is 2. You gained 2 ATP. I've said this over and over again. If you're a bacterium, that's fine. You're not, that is not fine. Now we have to take the pyruvic acid, we got to put it inside the mitochondria. But it has to change. And the change that it undergoes is that it has to be converted into this molecule called acetyl-CoA. Now that's inside the mitochondria. In the process of converting it to acetyl-CoA, you get carbon dioxide, and you get another substance. This other substance is our electron carriers. These electron carriers are what are known as NADH, or you're going to see NADH plus H plus. Suffice to say, just think of those as the carriers of electrons. These electrons have a lot of energy in them, and it's the energy of those electrons that are going to be used later on to generate ATP. So you want to capture that energy, and it turns out that NAD is a molecule that can hang on to electrons and transport them into the electron transport chain, which generates ATP. That is the big plus of all of these other processes that go on. So you see in glycolysis you get a little bit of high energy electrons. High energy electrons means those are going to be transported primarily by NAD, taken to the electron transport chain, generate ATP. The conversion of pyruvic acid to acetyl-CoA also does the same thing. You get some high energy electrons. And then if you go through the citric acid cycle, you also get high energy electrons. So these sources of these high energy electrons are taken to the electron transport chain down here. Okay? That's the importance of the aerobic pathways. They generate so many of these high energy electrons. And those high energy electrons are transported in, I always say, electron baskets, right? Imagine you're a bee and you're going from flower to flower to flower and you're gathering pollen. That's not really what you want to do as a bee, but you are gathering pollen, and you take the pollen to one flower to another flower, and that gets those flowers to reproduce, right? Well, as that bee, as the same idea, your electrons are getting stuck to your electron carriers as a bee, and then you're taking them to the electron transport chain. I get that the analogy loses something, but these high energy electrons are transported to the electron transport chain. And this is where you're going to get the vast majority of ATP, without a doubt. So let's do a little quick math before I move on. All right, glycolysis gives you 2 ATP. Okay, so you would say per molecule of glucose, for each molecule of glucose, you get 2 ATP out of glycolysis, a net gain of 2 ATP. Okay? When we go through the citric acid cycle, what do we get out of that? We get a net gain of 2 ATP out of that too. So 2 plus 2, you got 4 so far. All right? Electron transport chain, 26 to 28 ATP. Way, way, way more ATP out of electron transport than any of these other processes. And that's why you need to breathe. And breathing, oxygen turns out to be what we call the terminal electron carrier. And we're going to get to that next step. All right, so cellular respiration, here is the overall idea. We start with glucose. And in order to go through all of these processes, you need glucose and oxygen. That's why you breathe. That's why you have a cardiovascular system. I went through all that before. In the end, what do you get? ATP. That's really what you want. You get some heat out of it. And you get water and carbon dioxide. Now these are considered waste products, right? Carbon dioxide for sure is a waste product because you breathe it out. Water, it's not so much a waste product. I mean, you can use water elsewhere in your body, but if you don't need the water, you can eliminate it through your kidneys. And so it goes through an aerobic part and an anaerobic part. I've already told you this, anaerobics make little ATP, the aerobic part makes most of the ATP. Here's glycolysis in words. So we've been through some pictures already. But this is how glycolysis works in words. It's a series of 10 reactions. We're not going to go through all 10. Don't worry about that. But we take glucose, a 6-carbon sugar, monosaccharide. We break it into 2 3-carbon molecules called pyruvic acid. This occurs in the cytosol. I guarantee you that will be a question. Where does this happen? This happens in the cytosol. It is anaerobic, does not require oxygen, doesn't use oxygen at all. Gives you 2 molecules per molecule of glucose. That is the net overall gain. There are 3 phases to glycolysis. As I said, you have to put ATP into that reaction. You put 2 ATP in to get it started, to get glycolysis started. In the end, you get 4 ATP out. So 4 minus 2, you gain 2 ATP by doing all of this stuff. You also get the 2 3-carbon molecules. You already know what that is, pyruvic acid. And you get this stuff, NADH. I told you NADH is your electron carriers. Those are baskets of high energy electrons. Here's the process. Now, all I really care that you know is you start with glucose. Glucose is phosphorylated. That just simply means you're using ATP. You use 2 ATP to get the whole thing started. I don't care that you know that you end up with fructose 1,6-diphosphate. That's not important to me. What is important to me is in the end, you get 2 pyruvic acids. You get 4 ATP. You put 2 in up here. You get 4 out. You gained 2. Okay? You gained NADH plus H+. These are electron carriers. These are electron baskets. High energy electrons. Now, from here, you got 2 choices. If you have plenty of oxygen, you go into the matrix of the mitochondria. If you do not have plenty of oxygen, you generate lactic acid. You don't get very much ATP out of that. But it is a way to continue the metabolic process. Right, so now so this is anaerobic. Let's move on with the aerobic. These are the anaerobic. We'll summarize the anaerobic reactions. Now, I've emphasized this here. It says in the presence of O2. Of course, that would be an aerobic reaction. In the presence of O2, NADH and H+ deliver electrons to the electron transport chain with molecular oxygen as the final electron acceptor. You're going to see what that means in a moment. If you're talking about anaerobics, you're talking about in the absence of O2. That is in the absence of O2. There is no final electron acceptor. That's what oxygen is. That's why you breathe. I mean, literally, that's why you breathe. You breathe because you need some place for these high energy electrons to finally go, and that is with oxygen. And those electrons with oxygen form water in the end. But if you don't have that, you don't have enough oxygen, NADH and H plus deliver electrons and H plus back to pyruvic acid, and that's what forms lactic acid. Now the chemistry of that, I don't really care that you know that. I just want you to know that if you don't have oxygen, you're going to generate lactic acid, and that is the lactic acid burn. That's that muscle burn sensation that you get. What happens then? Well, lactic acid inhibits glycolysis. So now you're going to slow down even the production of even the tiny little bit of ATP that you have. What this means is if you run fast, so I always say if you're running away from a bear. I don't know why people run from bears. I mean, I get why you would run from a bear, but if you're running away, if this bear wants to eat you for lunch, you're going to run. You start running, okay? As you run, you can't possibly deliver enough oxygen to your working muscles fast enough for a long period of time. So at some point, you're going to start to run out of oxygen. That means you're going to start to generate lactic acid. And you're going to start to feel the burn. You're going to breathe faster, but you can't keep going. Lactic acid inhibits glycolysis. So now the little bit of energy you were making, even that slows down. ATP production is decreased. Glycolysis produces much less ATP than aerobic respiration. Way, way, way less. And overall, there is a net gain of 2 ATP per molecule of glucose broken down. You need more than that, so you got to go on to aerobic reactions. Here they are, aerobics. In the presence of oxygen, that's what aerobic means. Quite literally, in the presence of oxygen. Pyruvic acid enters the aerobic pathways, as you saw in that picture. The aerobic reactions include the synthesis of acetyl coenzyme A. Pyruvic acid cannot enter into the mitochondria by itself. It has to be changed. What's it changed into? This stuff, acetyl coenzyme A. Then that stuff enters the citric acid cycle, and then you generate a whole bunch of high energy electrons from that. Those high energy electrons go to the electron transport chain. We're not going to get into all the details of the electron transport chain. Just understand that you need electrons that go to the electron transport chain, and that chain produces out of those high energy electrons a lot of ATP. Right? So pyruvic acid moves from the cytosol into the mitochondria. Pyruvic acid is used to produce acetyl-CoA. In the end, what do you get? Your end products, your waste products in a sense, are carbon dioxide, water, and up to 36 ATP per molecule of glucose. So remember, the anaerobics gave you 2 molecules of glucose. The aerobic reactions give you 36 molecules of glucose. Way more efficient. This is the process that you want to go. This is why you breathe. Now here it is in picture form again, where you ought to know you get pyruvic acid from glycolysis. You already went through that. All right? Pyruvic acid goes in and is changed. It's changed into acetyl-CoA, coenzyme A. That's the stuff that can enter the citric acid cycle. Citric acid cycle, round you go. What you really get out of the citric acid cycle is you get a little bit of ATP, but not much. So the point of the citric acid cycle is not to make ATP. Now you get a little. But that's really not the point. The point here is you get 8 hydrogen atoms are transferred to NAD plus and FAD2. Or FAD, sorry. So just think of NAD plus and FAD as electron carriers. These are substances that can capture the energy of a high energy electron and transfer it to the electron transport chain. That's where you're going to get massive amounts of ATP. So it doesn't matter to me that you know all of the byproducts and substrates and all that kind of stuff. All I care about here is that it's called the citric acid cycle because the first component that is created out of the cycle, the first thing is called citric acid. Right? So citric acid is the first component. This cycle is also called the Krebs cycle because the guy who discovered this thing back in the 1930s or so, his name was Dr. Krebs. Hans Krebs. So he discovered this, so he's lent his name to it. Well, now we've decided to call it the citric acid cycle. It is also, on rare occasions, but you may also see it referred to as the tricarboxylic acid cycle, the TCA. So, yes, it has those three names. The three names are this. Same thing. There is no difference between the citric acid cycle, the Krebs cycle, or the tricarboxylic acid cycle. Same thing. In the end, you're getting a tiny little bit of ATP, you're getting some carbon dioxide, and you're getting electron carriers, right? And those are the important things. Now, we take those electrons, those high energy electrons, right? I always say they're in baskets. I don't know why they're in baskets. They're wiggling around, they're crazy, and you got to hang on to them. What the electron transport chain does is it takes NADH and FADH2, so these are compounds that have those electrons. When they have these electrons, these are referred to as reduced compounds. So NAD plus, before it has the electrons, is referred to as an oxidized compound. There's a way to remember what you call a compound depending on its state of oxidation or reduction. And something that is carrying electrons has been said to have been reduced, chemically reduced. In chemistry, we refer to that as a reduction, or it's been reduced. And if it's not carrying electrons, it's referred to as having been oxidized. Right? So we would say, and the way to remember this, one way to remember this is there's an acronym that's applied to this. And the acronym is OIL-RIG. OIL, and the second word is RIG. OIL-RIG. OIL stands for oxidation is loss. Loss of electrons. If you lose electrons, you are oxidized. But the RIG part is RIG. Reduction is gain. So, this compound, NADH, has gained electrons. And FADH2, we say, has gained electrons. So if they have gained electrons, they have been reduced. That's the chemical term. Right? So these reduced compounds are transported to the electron transport chain, and their electrons are handed off to compounds in a specific order. And they're handed off in a specific order because you can take each step in the electron transport chain can take a little bit of that energy from the electron and create and make ATP out of that energy. Right? So here's a high energy electron. It gives off a little energy. That energy makes ATP. And now we take a little bit more energy off, and that makes more ATP. And we take a little bit more energy off, and that makes more ATP. So the energy is extracted in tiny amounts, and each tiny amount makes more ATP. And that is the point of the chain. At each step, at each phase, you can take the energy off in small doses rather than a big bomb, as I told you with sodium or with an atomic bomb, right? You get all of the energy out all at one time. Kaboom! Here, you get the energy out little tiny bits at a time. That's the whole point of the electron transport chain. So you take these compounds that have gathered up their electrons from other parts, some of them from the anaerobic part, but most of them are from the aerobic reactions. You have a bunch of reduced compounds that are carrying electrons. They bring it to the electron transport chain. They hand the electrons off, and the energy is extracted in small amounts to make ATP. That's it. But in the end, you still have electrons left over. These electrons that are left over still have some energy, but not enough to make more ATP. Well, you have to do something with those electrons. What do? You combine them with oxygen. If you combine them with oxygen, then you get H2O. Now, we don't have to worry about the chemistry here. That's not really the important part. The important part is you still have the electrons. You've got to do something with the electrons. What do? You combine them with oxygen, and you make water. That's why you breathe. I mean, literally, that's why you breathe. Right here. If you do not have oxygen here as the final electron acceptor, you don't get the water, but you can't run through the electron transport chain. Now, I bring that up because there are certain poisons that can stop the electron transport chain. Cyanide is one of them. Cyanide interrupts the flow of transporting the electron from one compound to the next compound to the next compound. It stops that chain. And so even if you are breathing, you're still losing energy, and you're going to die. That's what cyanide does. It hits right in this electron transport chain, and it is a poison that stops this chain, and you can't make ATP, and so you run out of ATP. Well, if you run out of ATP, you're dead. And that's all there is to that. All right, so big picture. Here it is. Big picture again. Glucose goes through glycolysis in the cytosol. Here's your review. Right? 6-carbon compound, 6-carbon monosaccharide. You get 2 3-carbon compounds called pyruvic acid. And you get 2 ATP, a net gain of 2 ATP. All right? If you continue with oxygen, now the aerobic reactions, that requires pyruvic acid to enter the matrix of the mitochondria, where it has to be converted into acetyl-CoA. When that happens, acetyl-CoA can now enter the citric acid cycle and go round and round and round and round and round. And from the citric acid cycle, you get a tiny little bit of ATP. You get some carbon dioxide, but you also get more high-energy electrons. Now, these high-energy electrons that are created in all these places come down, and their energy is extracted tiny little bits at a time, and that energy is converted to ATP. As long as you have oxygen present. No oxygen, or not enough oxygen, things stop here, and pyruvic acid is converted into lactic acid. If you have plenty of oxygen, things go into the matrix of the mitochondria. You run through all these processes, and you end up with a lot more ATP and water and carbon dioxide as waste products. That's it. In the end, if you go through glycolysis only, you get a net gain of 2 ATP. Right? If you go through the whole electron transport chain, you get 36 ATP. So you're talking 18 times more efficient, 18 times more ATP going through the aerobic reactions versus just simply the anaerobic reactions. You have to understand that's why you breathe. That's the importance of oxygen. This is where it all happens. This is where your body gets energy to do anything. You want to move around, your muscles need ATP. You want to think, your brain cells need ATP. You want to filter your blood through your kidney, those cells need ATP. You want to beat a heart, you need ATP. ATP is cellular money to do everything. Right, so where do you get these sources of energy? Have to be converted, right? You don't eat ATP, you eat bread, or you eat vegetables, or you eat meat, or you eat, whatever your diet consists of, whatever you put in your mouth has to be broken down, and it has to be turned into ATP. Now we go back to remember the first week of lectures, right? We have catabolic pathways that produce energy, so anything that you eat has to be broken down, and ultimately broken down to the stuff that you need. In this particular lecture, in this case, we're talking about ATP. But you're going to take that energy from the stuff that you break down and you're going to put it into life functions. You're going to make more proteins. You're going to repair your DNA. You're going to filter your blood. You're going to pump your heart. You're going to think, whatever you're doing, right? In order to build things up or to fix things that you broke. You know, you sliced your thumb while you were cutting the bagel. Well, you got to reproduce those skin cells. You got to fix those damaged blood vessels. You need to do that through anabolic pathways. So catabolic and anabolic pathways work hand in hand. They both need energy, though. You get the energy from the catabolic pathways to do anabolic things. If you eat more food than you need, you can store some energy in various forms. In fact, you can really store it in a couple of different forms. So you eat carbs. We're gonna say you start with monosaccharides. Now, yes, there are other sources of food. We're not gonna. The next slide will kind of get into that a little bit. But for our purposes, all we really care about. We got monosaccharides. What are you going to do with them? If you need them for energy right now to do stuff, okay, great. You can do that. You're going to put them through catabolic pathways. You're going to get your energy, ATP. You're going to get carbon dioxide in the deal. You got to breathe out, and you're going to get water. We just went through that. On the other hand, if you don't need the energy right now, you can store those substances. So monosaccharides can be stored in the form of glycogen, and I showed you that in that picture slide that I have. Glycogen can be stored in muscles or predominantly in the liver as glycogen. You can convert monosaccharides into triglycerides, which are lipids, which are fatty substances, and there are ways to do that. Or you can create amino acids if you need amino acids to make muscle or to make other proteins, enzymes, whatever you might need. You can convert those to these other substances. So you can go through catabolic pathways or anabolic pathways. As I say, glycogen, most cells have a little bit of glycogen. The liver and muscle cells store the vast majority of glycogen. If you really don't need it and you really need long-term storage, you can convert these things to fat. Fat storage is a very long-term storage, a future energy source if you need it. Now, here's the summary slide, right? Here's somebody who ate. They ate some toast, they ate some butter on it, they had some eggs, whatever. They had some, eggs and I don't know if they're ham over there. I don't know what they had. Anyway, they ate something. The carbs, we just went through that. Carbs are broken down to simple sugars. The simple sugars go into glycolysis right away. Boom. Pyruvic acid, acetyl-CoA, citric acid cycle, electron transport, boom, 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 H2O, and CO2. Great. We just went through that. You can eat butter. Let's say butter is a fat. Fat can be converted into glycerol, and that can enter into this whole chain of command. Don't expect you to know this. You can also get, out of the fats, you get fatty acids. Fatty acids can also enter into this whole metabolic process, pathway and process. And then amino acids, right? Proteins are broken down to amino acids. This is what digestion does. It breaks it down to amino acids, or breaks it down to simple sugars, or breaks it down to glycerol and fatty acids. If you're going to extract any energy from anything, you got to break it down. That's what digestion is all about. Once you've digested it, then you can do different things with those substances, and they can enter into this pathway at various points. As I say, if you take an advanced biochemistry class, you're going to get into this more. If you go on to, higher medical courses, you're also going to have to learn all about this kind of stuff. But we start with carbohydrates, because that's the preferred and the easiest source of energy for your cells. Glycolysis is number 1. Citric acid cycle is number 2. Electron transport chain is number 3. And that is combined with the other process called oxidative phosphorylation. That just simply means making ATP. And I believe that is it. Yes. There's the show. There's the ideas. That's what I mean when I say bioenergetics. Bioenergetics is the process by which we extract energy, convert it chemically to the kind of energy that we can use in cells, and that is ATP. And we use that energy to do everything. But we have to constantly recreate, regenerate those sources of energy all the time. Thanks for your attention. That's it for bioenergetics. I will be communicating more with you in the future and more topics. Thanks.